because with all of you in this room, who I'm very grateful to you all for showing up, it's a phenomenal opportunity for Global DARE Foundation to have so many highly invested researchers and clinicians in the same room, discussing all of these points and helping us identify a path forward to help all of the patients that are also in the room. So thank you. To get started, I was very pleased to be part of each of the work groups that we had, and there were definitely some interesting takeaways from these work groups. So moving to the next slide. What I did is I decided that it might be helpful to sort of break up this presentation into 3 different parts. One was the insights that I was fortunate to take away with, and then the shared infrastructure that obviously is so apparent that we need to start putting in place. And then identifying the concrete next steps, like what can we do, not necessarily 5 years from now, but really starting tomorrow, to start making progress towards the infrastructure, the trial readiness, like all of those milestones that we need to have in place to make progress. So for the first part, what we learned that wasn't really obvious at the start. Obviously, every work group that we had all knew that we needed longitudinal studies. We all knew that we needed patient outcomes and biomarkers because this is something consistent across every discipline. Every type of disorder, every disease needs to have a way to measure how patients will improve over time with a therapy. And then what infrastructure is needed to act on those understandings or learnings that we had. And then what we can, as a group, hopefully kind of commit to building after this meeting today. So what did we learn? So at least for me, these are my slides, so these were my learnings, I guess. But the biggest obstacles weren't really the therapies themselves. We have a plethora of opportunities, I think. And so I think the patient should feel very pleased with the level of progress that's been made in the field. Not just small molecules, not just gene therapy, not just ASOs, but all of them moving together forward together, rather. I think the biggest obstacle for us are really our gaps in understanding the disease itself. In addition, obviously, part of that is measuring the meaningful changes that we see if we were to introduce a therapy, and evaluating the benefit of each of those interventions, and possibly also not just the benefit, but also if there are disadvantages, or somehow worsening a situation that already exists. And then I think also learning from patients that are already receiving care is an area that we need to start to hone in on and sort of capitalize on if possible. So in terms of the learnings, we understand that there's a metabolic effect. We understand, obviously, that there's a Phi-H deficiency, and this causes an accumulation of phytanic acid. And we also know that lowering this level of phytanic acid still remains sort of the cornerstone of treatment for adult refsum patients. What we don't know is why there are certain tissues that are selectively affected. Is it where the lipids are being stored, or are certain cells actually more susceptible to damage than others? Why disease severity varies between patients, and also how phytanic acid ultimately causes the tissue injury that we see. So the roadmap implications here are really measuring the phytanic acid alone, it's not enough. We really have to understand the disease mechanisms that are driving the tissue injury that we're seeing. And of course, I'm preaching to the choir, all of you know this. So for critical research questions, for us, why are certain tissues selectively vulnerable? I think, there's been several patients who have identified that, for them, their biggest concern is the loss of vision. So obviously, our concern then is identifying why the retinal cells are so susceptible to this increased level or accumulation of phytanic acid in the system. Which forms of phytanic acid are biologically active? So Anne has, beautifully shown in her work that you can store the triglycerides in a relatively stable form, but if it's stored in a mitochondrial membrane or retinal pigment epithelium as a phospholipid membrane protein, obviously this is going to cause other types of effects. And then what cellular mechanisms are actually driving the tissue and the disease progression? So from my perspective, it's not just what therapy we need to start getting a handle of. We really need to understand the lipid biology, of which we're very grateful for all of KKI's support, and also any kind of mitochondrial dysfunction. Oxidative stress potentially may play some role that we have yet to identify, and understand better the neurodegenerative pathways that are occurring. The roadmap implications here are obviously understanding disease mechanisms require these biomarkers, as well as the mechanistic studies. Learning too is we really cannot yet define success well enough. And I think there's a problem with this too, is I think success for each patient is going to look different. Someone who is more advanced with less vision is going to have a different idea of what success looks like than someone who hasn't yet started to lose their vision, but knows that they have 2 Phi-H mutations. And I think Florian had mentioned this, if we did have a therapy that lowered phytanic acid tomorrow, would patients be able to maintain their vision? Would they be able to do all of these things that they want to do? And so we don't know, because we don't know the disease. And so here the roadmap implications were we need outcome measures and patient-reported outcomes in order to really move our work forward as a foundation and to support your research. And then for me, I guess learning 3 was, I felt that apheresis represents a unique opportunity to learn more about the disease progression. We have an interesting opportunity with German groups or other groups throughout Europe who are going in every week or every other week, and they're having their phytanic acid levels taken, and some of them are doing vision tests. But beyond that, their quality of life really isn't being measured. Their ataxia or other types of progression metrics aren't being put in place. And when I asked Dr. Klingel this, I think I made a reference to the German cohort of apheresis patients, and he was good enough to correct me that it's not a cohort. These people aren't being studied. They don't have a single type of therapy that they're undergoing. A lot of them are having either acute therapy, they're having chronic therapy, they're having plasma exchange, they're having the lipid apheresis. And so I feel like this is an excellent opportunity if we can sort of identify sort of a way to, again, insert metrics that could be standardized or harmonized across at least this population that is regularly undergoing treatment currently for adult refsum disease, that we could better understand more about the disease and how it progresses. So apheresis is interesting because it started in the late 1970s as a treatment for adult refsum patients. And so yet even 50 years later, we still don't know who benefits from this treatment. We don't know when the treatment should begin or how often it should happen or what approach is optimal. And so Germany, I understand, does have an apheresis registry of 5,000 individuals, but only 1,000 of those are actually ever annotated in a meaningful way. And so, perhaps even working to, get physicians more involved in annotating those patients and monitoring them in a meaningful way so that we have time points that we can look at over the course of intervention for apheresis. So I think the roadmap implications here were we need to systematically learn from the treatments already being used before we start applying others. And so what does this mean for therapeutic development? So I think the work groups identified a lot of very realistic therapeutic directions. However, regardless of the intervention that they were hoping to further, everyone came down to the same foundational capabilities of understanding the disease biology. And so, again, the roadmap implications here was building research infrastructure and how that will really impact our ability to make and understand successful therapeutic studies. So infrastructure required. So basically, I took each of my learnings and I tried to understand exactly in terms of infrastructure what would be required. So obviously, the disease mechanisms remaining incompletely understood, we need more mechanistic research. The natural history study is wonderful, and we will be moving forward with it. But in terms of identifying on a fine scale basis, I think we're going to have to be in a position, obviously, where we can utilize our research assets more, to wield them in a more specific kind of way, to really understand how the disease progresses over time and what points in time we can intervene in a way that makes sense therapeutically. Again, meaningful clinical outcomes, we need to identify the clinical outcomes. And then current clinical experience is not systematically captured. And so this comes down to harmonizing clinical data sets right now for adult refsum patients. And I realize it's something very easy to say, but with all of these patients sort of geographically dispersed throughout the world, it becomes difficult with insurance coverage and access to care and so many other issues. But it is something that if we can even find a very simple, harmonized data set, something with just a few metrics that we could begin to follow patients, I think over time we could grow that organically as needed. Again, I'm always one to sort of harp on things, so I always repeat my slides. So infrastructure priority, again, natural history and longitudinal monitoring. This is needed, absolutely, in order to understand how ARD progresses and what changes occur over time. And then for the implementation of harmonized data collection. For biomarkers and outcomes, I feel, especially going into this next conversation, we've talked about some of the potential biomarkers. Joe, in his talk, mentioned 3-methyladipic as a measure for omega oxidation. Also, 2-phytanic acid, which unfortunately isn't accessible to everyone, but if we had a dried blood spot analysis might be, far more accessible. But also other measures, things like the ratio of different cholesterol phytanyl triglycerides that I know has been reported in the past, as well as potentially mitochondrial stress or oxidative damage markers. And obviously, for the purpose of disease progression and for the purpose of reflecting treatment response, these are invaluable. And so we're hoping that we can identify a small core data set that we can utilize for this in both outcomes as well as understanding biomarkers and consistently having those measures over time. So what can we start building tomorrow? So the idea is not just to hold these work groups separately and move on. It's really to identify what the next action item is. And so can we potentially generate an apheresis data set? And if so, what would that look like? Right now, I believe that Cordula and Reinhardt are collecting vision data from each of those points as well as phytanic acid. Are there other things that they could be testing? Could they be testing for neurofilament light chain? Could they be testing for markers for oxidative stress? Could they be testing for whatever biomarkers that you feel might be worthwhile? Also, core biomarker panel. In terms of future studies, is it possible to, even today or in the next few months, work to agree on a few things that we can begin measuring in a standardized way, just to start, just to sort of put our foot, in the door and start the process. And then also core outcomes. Especially with vision and neurology, are there 3 to 5 outcomes, we could do fewer, that we can capture collectively across our populations, and work towards a harmonized data set in that way. And so these are all things that we would love to be able to sort of to work towards, to start to implement. And so I wondered if we could even have a discussion about making a commitment before we leave today about what could be done and how. And I realize that, obviously all of these things take funding. And so if funding were not an option, an issue, what could we begin doing today or tomorrow that would really begin the process of moving towards trial readiness for the adult refsum population? And so, in terms of an apheresis data set or a biomarker panel, outcome measures, or anything else, even mapping out all of the research assets, things that would make it more accessible to do the research necessary to understand the mechanistic aspects of adult refsum disease. So I'm going to leave it here so that you can think about it. And maybe with that, I'll take questions or comments as to how you feel would be the best approach.